So our first presenter for insights into T1D pathogenesis immune perspective is Dr. Dan Moore. I'm just going to give a brief introduction to this series. We are talking now sort of the counterpart of the insights into T1D pathogenesis, the islet side. Now we're talking about the immune perspective. And so the first speaker is Dan Moore. He's an MD, PhD. He's Associate Professor of Pediatrics and Pediatric Endocrinology and the Associate Professor of Pathology, Microbiology, and Immunology here at Vanderbilt. Then followed by him, we're going to be hearing from Dudley McNitt, a PhD, postdoctoral fellow, research fellow in the lab of Dr. Rachel Bonamy in rheumatology and immunology, followed by Chris Wilson, instructor in pediatrics and pediatric endocrinology, and a word from Dr. Al Powers as well.
Thanks for having us on and for visiting Vanderbilt. So I'm going to talk a little bit about some translational science and what we can learn about the pancreas from imaging it. So islets are very small and scarce, so they're like less than 1% of the pancreas. You basically never see them in any kind of study. But the question lingers, like, what could you learn about the biology of type 1 diabetes from longitudinal studies of the whole pancreas, particularly in people that still have their pancreas? And are there changes in the pancreas that can be measured and relate to the timing of disease? I may have seen this slide or image during the day, but the timing of disease is long from the initial detection of autoantibodies to loss of beta cell secretory function and presentation in the clinic with type 1 diabetes doesn't happen until like years to decades after the diabetes has started. So we developed an imaging pipeline here at Vanderbilt about a decade ago, and we've had support from a number of different places to get this done. We've had a number of people recruiting patients. We place them in a nice regular old MRI machine with all the clunky whirring sounds. We have a wonderful radiologist that outlines the pancreas. Melissa Hilmes is probably the most knowledgeable person in the world about the shape of the pancreas at this point. Probably she's close to 10,000 pancreases outlined. And then Dr. John Verasco, who was originally at Vanderbilt and is now at UT Austin, does the image analysis. And we also collaborate with individuals at the University of Barbara Davis Center in Colorado, with the St. Vincent's Institute in Melbourne, Australia, and with the University of Chicago to do these studies. We spent a lot of time at the beginning standardizing measures of the pancreas. We want to make sure that when we do imaging, when we study you, that we get the same result every time. So Dr. Verasco developed what are called phantoms, which are basically fixed measures that you can put in a machine and they should measure the same way every time. He also created a 3D printout of his own personal pancreas that's been shipped like all over the world at this point, been measured over and over again to make sure you get the same size every time. So his pancreas is the most well-traveled imaged pancreas in the world. And these went all over the country and the world, and also healthy volunteers went all over the United States to make sure that we get the same results every time. And so then we were able to study people with type 1 diabetes and healthy controls. And this is a wonderful example of what we've learned. So remarkably, these are two 10-year-old boys. One's healthy, one has type 1 diabetes. They both weigh exactly 64 pounds. It's very remarkable. I guess you do enough imaging, though, you can find exact matches of each other. And the pancreas in the individual type 1 diabetes is half the size. So this person, the 10-year-old young boy, has had new onset type 1 diabetes within the first 100 days he was imaged. His pancreas at the diagnosis of diabetes is half the regular size. And you can see it's like kind of scrawny, it's thin, it's elongated, doesn't have this nice sort of fat chunky shape of the regular pancreas. And we've repeated this over and over. So not just this one image, but we've done a lot of healthy controls and other individuals. It's a pretty simple study. It doesn't require any contrast. It takes about 25 seconds to get a really good picture of your pancreas, but then we do some more imaging to try to get some more details. And you can see people with type 1 diabetes, the new onsets, first 100 days after diagnosis, their pancreas volume, here represented as their pancreas volume index, which is their volume divided by their weight, is about half that of healthy, similar individuals. So some of the people with type 1 diabetes have more normal sized pancreases. Almost none of them have like bigger than average size pancreases for healthy people. And a lot of them have pancreases that are smaller than anybody in the healthy population. So that suggests that there's something that can be measured in people with type 1 diabetes. Pancreas volume continues to decline. It's an active process. There are things happening to you even after the diagnosis, that being stabilized, having your blood sugar improve, starting on insulin therapy doesn't stop from happening. So the pancreas in the first year continues to decline at a pretty good clip, and then it just sort of lingers its little constant rate of decline for the first 5 years after diagnosis. These are individuals we've had come back and imaged over and over. Each line is a separate individual that's participated in the study. So this is a longitudinal study of individuals with new onset diabetes. And then, you can study people forever. Well, certainly, I guess you can, but we haven't had enough time. But we're curious about what happens to the pancreas in longstanding type 1 diabetes. And so now we've got people that have had diabetes for decades, 10, 20, 30, 40, 50 years. And no matter how far along you are in life, no matter how long you've had diabetes, the further along you get, the smaller your pancreas is relative to healthy controls. Generally, healthy people are trudging along, keeping their pancreas about the same size per their body weight throughout life, about 1 milliliter of pancreas volume for every kilogram of body weight that you have. Well, if you have type 1 diabetes, you're shrinkifying, shrink every decade that you continue with the illness. So we were really curious though, like what about before diabetes? Like what is happening in the time leading up to the diagnosis? So we're fortunate at Vanderbilt and at our collaborating centers that we're part of the TrialNet program that has been following people for the risk of developing type 1 diabetes for many, many years. It identifies people, it studies people that are relatives of individuals with type 1 diabetes. It screens them for autoantibodies, and then it identifies people that are multiple autoantibody positive. You probably know if you're multiple autoantibody positive now, meaning 2 or more, that you have stage 1 type 1 diabetes. And it's just a matter of time until you get type 1, until you get insulin requiring type 1. You have it already, but you might not need shots for a while. And so one of the questions would be like, well, how long is it going to take? How long is it going to take for me personally to progress to stage 3 type 1 diabetes? And we were hopeful that maybe this changing dynamics of the pancreas could give some insight into that. We found very interestingly that based on how big your pancreas was at the beginning of enrollment, like when we first met you, when you were at stage 1 or stage 2, bigger pancreas was protective against developing stage 3 quickly. Smaller pancreases got to stage 3 first. These little stars were the very first moment when someone developed stage 3 type 1 diabetes. But you can see pretty clearly the smaller your pancreas is, the more at risk you are of developing the next stage, often within a year or a year and a half of our meeting you. In your early stages. If your pancreas was small, you're off to the races. Whereas if your pancreas was bigger, you're making it out here 3, 4, 5 years, and you're not yet progressing to symptomatic stage 3 type 1. Interestingly, from a scientific perspective, measures of the pancreas were independent of metabolic measures. There's a lot of common metabolic measures that people make. People get oral glucose tolerance tests, and those can be derived into metrics about pancreas function. The index 60 and the diabetes risk score are 2 common metabolic measures, but the pancreas volume index didn't really predict those. When it predicts, they would line up as a line. The 2 metabolic measures predict each other very, very well, but pancreas volume index adds a whole new wrinkle and whole new information to the risk of developing stage 3 type 1 diabetes. You can also see in the graph here that the loss of pancreas volume begins years before diagnosis. Time 0 here is the time at which these people showed up with stage 3, that had stage 3 by a glucose tolerance test measure. And you can see even up to 4 years before the diagnosis, the pancreas volume is starting to slip. On the other hand, with the metabolic measures, they begin to shoot up as you're closer to diabetes, and the shooting up is well known to happen about 18 months before diagnosis. You can see that here, suggesting that the pancreas volume loss may be the earliest sign that you're about to develop stage 3 type 1 diabetes. And if you knew that, you might be able to access therapies earlier. You might do closer monitoring of yourself for your risk of developing stage 3. You might be more protected from DKA. Information about your risk of progressing is very valuable to you as an individual and to studies broadly. So the pancreas size really does predict your risk of developing stage 3 type 1 diabetes. If your pancreas volume is small, you're on the fast track to developing stage 3. And if your pancreas is bigger, you're relatively protected from developing stage 3 over the next 3, 4, 5, or 6 years. This is really helpful to know in terms of developing new clinical studies and interventions. The question remains as to why. Why is it that your pancreas is shrinking when you're progressing to stage 3 type 1 diabetes? And we've had a lot of opportunities to learn clues as to why this is. Our collaboration with the University of Chicago and their monogenic diabetes registry has been really helpful. So these are cases of diabetes where one gene causes you to develop diabetes, abnormal blood sugars. In this case, the insulin gene was mutated. And this family, this wonderful family who gave us their photograph to use, they had both affected and unaffected family members. Some people had an insulin gene mutation, developed very early diabetes requiring insulin generally in the first year or even first 6 months of life, and other people were not affected. If you were unaffected, you had the regular gene, your pancreas volume index was basically that one, the normal size. If you had the mutation, you had an abnormal insulin gene, your pancreas size was very, very small. If your pancreas was small, you didn't make much C-peptide. If your pancreas was big, you did. If your pancreas was small and you had this mutation, your pancreas was long and thin, like we showed you earlier. But if you were the healthy family members, your pancreas was nice and chunky and good looking like a regular healthy individual, suggesting that pancreas volume is strongly related to your insulin secretory capacity. So pancreas volume index for us is very exciting. It's the newest feature that can predict diabetes stage progression. This is really critical for decisions about treatments and designing clinical trials to make them have the fastest decisions. If you know who's getting diabetes next, it's in your best interest to help them try to delay that. It appears to be independent of any other measure that we know about and adds predictive value. Changes in volume are also apparent prior to any other measure, so it goes first. And it appears tied to the most important thing of all, which is your loss or capacity for insulin secretion. So we're very excited about all that. We look forward to incorporating into clinical studies and trials and to using it to help us make better decisions and better understand the biology of diabetes.
Thank you so much. That was great. Have you done future studies after on any tissues that you've been able to obtain postmortem and seen whether or not the ratio of endocrine to exocrine changes significantly as the pancreas as a whole shrinks?
Yeah, so it's a little bit hard to ascertain because as the pancreas is gone, like the islet surrounding areas, like the ratio still seems okay, but the whole pancreas as a whole is smaller. So suggesting what I think happens, although we don't know, is like you lose this islet or this islet becomes non-functional, the insulin doesn't flow out, and then the surrounding tissue atrophies and that whole little area just disappears. But more studies are needed to know for sure. And of course, postmortem studies only are one snapshot in time. Our longitudinal studies like see things happening over time, but you don't get samples of the pancreas. There's a classic study in Japan, the DIVID study, where they did sample people, but it was had so many complications after having your pancreas poked that I don't think anyone will ever get to do that again.
Here's a question from Ian Sweet. If changes in pancreas size occur early, but is correlated with insulin secretion loss, wouldn't they both occur at the same time?
Yeah, so what I think happens, but it's only more study, is your insulin secretion that you measure in the periphery is the integration of all the little islets dumping into the circulation. And so it might still have enough to measure a normal amount peripherally, where you lost this one little one down here, and you can measure the consequence. So you lost these 5 little ones around the pancreas, and the pancreas shrunk a little bit that you can measure, but the integrated, pouring out of all the other islet buckets still is enough in the periphery.
All right, well, to try to keep on time track, we'll take the next talk starting. Thank you again. That was really fascinating. And we've had Jack Verosco walk through some of his data, which are really nice as well. Shout out to him if anyone wants to look in our catalog and watch that talk. He's some really nice imaging as well. So now we have Dr. Dudley McNitt. He's the postdoctoral research fellow in the Bonamy Lab, and he's going to share his work with us.
Great. Hi, everybody. I want to thank Monica and Sugar Science for giving us a chance to talk about our work today. And I'm here as a postdoc in Rachel's lab. Rachel's apologies that she couldn't make to this meeting, but I'm here to represent some of the work done on the human side and then some of the work that I'm doing on the mouse side. And we're really interested in our lab on B lymphocytes, germinal centers in type 1 diabetes, and again, using our access to human samples as well as our use of mouse models to get at the importance of both of these cells and these structures in T1D pathogenesis. So there we go. So as Dan highlighted some of this, I'll try to skim over, but we all know that type 1 diabetes is an autoimmune disease characterized by the destruction of insulin-producing beta cells in the pancreas. And while cytotoxic CD8 positive T cells are thought and are pretty well established to be the ultimate drivers of beta cell destruction, other immune cell subsets play important roles in the immunopathogenesis of type 1 diabetes, and such as anti-insulin B lymphocytes or B cells, but I'll try to refer to them as B lymphocytes, not confused with beta cells. But our lab and others have shown that in terms of the roles that B lymphocytes play in type 1 diabetes development, is they have two well-known functions: their ability to produce high-affinity autoantibodies to islet autoantigens such as insulin, and their ability to present autoantigen peptide to cognate CD4 positive autoreactive T cells. And both of these functions are highly associated with germinal center structures, microanatomical sites found in lymph node, and even can be found within inflamed tissues such as the pancreas, where B lymphocytes will signal and present antigen to CD4 positive T cells to drive the maturation of both of these cells and lead the production of antibodies and the maturation of CD4s. Now, regards to the antibody production, these antibodies are not thought to mediate tissue destruction, but they do serve as an important biomarker to predict the likelihood of developing type 1 diabetes, as Dan illustrated earlier. The more different islet antibodies you have, the greater likelihood you have of developing type 1. And what Rachel is interested in the lab, and this is work that I am around, but not necessarily work that I do, so I'll just give the most general of highlights, is these are rare cells to find in the periphery. And so some of the techniques we use in the lab to identify these cells is the use of RNA sequencing and B-cell receptor sequencing and these sort of single cell approaches to identify insulin binding B lymphocyte subsets in T1D. And we're very fortunate to be a TrialNet center here, and the access of having these TrialNet samples allows us to take at-risk T1D peripheral blood mononuclear cells, or PBMCs, as well as unaffected controls, isolate them and store them, and then eventually pull out these samples from patients at different stages of T1D development, sort the B cells via CD19 positivity, and then analyze these B cells from both groups via the 10x platform of RNA sequencing, BCR-seq, and we can also do site-seq. And when using the single-cell RNA sequencing, we saw that clonally expanded B lymphocytes in the pre-T1D individuals were mostly found in the memory subset of B cells, which was not observed in the unaffected controls. And I want to highlight here that some of the data we generate in single-cell RNA sequencing looks like these UMAT plots, which are a two-dimensional representation of multiplex or complex data. And each of these numbers represents a different cluster listed here on the right of different subsets of B cells, different types of B cells. And you can see in the blue box and circled in the blue circle here in the plot that these are defined by their transcription and surface receptor expression as being memory B cells, this 1, 3, and 7. And when looking in these same clusters and then looking to see which B cells which share the same BCR are expanded, we find that there's a greater degree of expansion in these memory subsets in the pre-T1D individuals relative to the unaffected controls. And so, and this data in relation, or rather in conjunction with previous work by our lab showing that anti-insulin B lymphocytes tend to skew towards a memory subset, led to the hypothesis, or one of the hypotheses in our lab, that clonally expanded B lymphocytes harbor insulin-binding B cells, or B lymphocytes. And to test this hypothesis, others in the labs, Lindsay Bass, a graduate student, and others highlighted at the end of my presentation, use this the ability to express these B cell receptors as monoclonal antibodies, or mAbs, via looking at the B cell receptor sequence and maybe modifying it, something I'll highlight more in a moment, synthesizing recombinantly the DNA and cloning it into mammalian expression of vectors, both the heavy and light chain, and then expressing these B cell receptors, purifying them, and then assessing their ability to bind insulin, one of the major islet autoantigens, in an insulin-binding ELISA shown here in the bottom right. If you can see my cursor, I'm also highlighting it. And what they found is that in these B cell subsets that are these memory B cell subsets, they do find insulin binding B cell receptor sequences within this population, suggesting that this population of B cells is an important population to be examining in the pre-symptomatic stage of type 1 diabetes, and as something to then investigate in future studies. But going back to the other function of B cells in type 1 diabetes, and more about the work that I've been doing in the lab, this other key function of B cells is their ability to present autoantigen peptides to CD4 positive T cells. And again, this function is associated with germinal centers, and the type of CD4 positive T cell that's highly associated with germinal centers, is important for germinal center formation, is known as a T follicular helper, or TFH. And these TFHs are highly associated with T1D like B cells, And we know from studies looking at new onset T1D patients relative to healthy controls that they have elevated levels of T follicular helper cells in their peripheral blood relative to unaffected controls. There are mouse models that use transgenic T cell receptor models of TFHs that show that you can transfer disease using these engineered systems, but we really don't know what TFHs are doing in type 1 diabetes. It's been theorized that the anti-insulin B lymphocytes may be licensing or activating these TFHs, and again, this is theorized, or that this activation then leads to these TFHs either within the lymph node or follicle, or to then leave the follicle or germinal center, maybe adopt a slightly different phenotype, but then go to the pancreas and support cytotoxic CD8 T cell functions via pro-inflammatory cytokines. And in my work, I've been really interested in stressing the importance of these TFHs in a spontaneous model, and are they important in T1D development. And one thing to note about TFHs to understand what I did is that TFH differentiation and maturation depends on the transcriptional repressor BCL6, shown here in this diagram. And I hypothesized that if I could eliminate BCL6 and CD4 positive cells in one of our mouse models, our NOD mice of type 1 diabetes, that this would disrupt TFH maturation and ultimately prevent type 1 diabetes development. And to do this, we use this CRE-LOX system where you can introduce LOX-P sites around the your gene of interest, in this case, parts of BCL6, and then using a CRE recombinase, excise those regions as essentially deleted gene. And in this case, we introduced the LOX-P sites around portions of BCL6 and then bred these mice to a mouse that expresses the CRE recombinase under the CD4 promoter. And throughout the next few slides, I'll just be referring to our control mice as BCL6 sufficient, shown in orange, or our experimental mice are BCL6 deficient mice, shown in purple. And one of the first things we did was a disease study where we monitored for type 1 diabetes development in these mice over the course of 40 weeks by checking their blood sugars once weekly. And what we found is that, as you can see, that without function, that BCL6 deficiency and the CD4 positive driven BCL6 deficiency specifically led to a significant reduction in type 1 diabetes development in our mice, near complete protection, and really complete protection in our cohort of female mice. And when looking at, okay, well, what about autoantibody production? When we look at anti-insulin IgG in these mice, comparing the BCL6 deficient on the left and BCL6 deficient on the right, there's a significant reduction in anti-insulin IgG in these mice, suggesting that germinal centers are impacted. And lastly, when using flow cytometry analysis and looking for the classic T follicular helper cell markers, those being CD45 positive, CD4 positive, FOXP3 negative, cells that are high for PD1 and CXCR5, these are representative plots from the pancreatic draining lymph nodes shown here on the left, and then the numeration of that data shown here on the right. We found in the pancreatic draining lymph nodes, as you can see in the representative plots from the sufficient on the left here and the deficient on the right, that there's a significant reduction in T follicular helper cells in these mice. In the PLNs, or the draining lymph nodes, an important site for immune activation and priming of the immune cells, but also in the pancreas. I didn't want to show this data for the sake of time, but we do see this loss in the pancreas, as well as a loss of germinal center B cells in these mice. So as a kind of our take homes, our lab's really interested in overall, what are B lymphocytes doing in type 1 diabetes via, or be it their ability of really looking at the B cell receptor sequences and understanding the mechanisms of how these anti-insulin B cell receptor sequences and anti-insulin B lymphocytes undergo activation, and how do they recognize insulin, and understanding how the B cell receptor changes over time and if it's maybe polyreactive, if there's somatic hypermutations, can inform us about how the B cells might be engaged and activated, and help inform clinical trial development and the successful use of therapies in the future as we continue on this research. Our lab is also very interested, obviously, in germinal centers, because both of these functions of B cells are associated germinal centers. And my work specifically, I think, highlighted that BCL6 is an important target in T1D, at least in a mouse model T1D, and that this needs further exploration and investigation, as there is some therapeutic or translational appeal here, as there are small molecule inhibitors of BCL6 that could be used to potentially have another tool in the toolkit at potentially using to prevent T1D development, maybe in patients or individuals that are, as Dan has described, that we can better characterize as being likely to develop type 1. And I very quickly just want to acknowledge there are many members of the Bonomi lab here that have helped with this work, both on the BCL6 mouse side and on the human side. Several members of a neighboring lab in our lab have helped us, Chrissy Brandon, for example, my former mentor, Tom Thomas here at Vanderbilt, who helped me with the BCL6 project, our numerous funding sources, and I'm happy to take any questions. Thank you.
Thank you, Dudley. That was some beautiful results with really a rescue from diabetes with the BCL removal. Quick couple of questions here in the chat. We've got from Katigurda, do memory B cell subsets have any correlation to time of progression of diabetes?
That's a good question. I will say I can't answer that fully because I'm more on the team, as I'll say the mouse side of the research, but I know that that's something that Lindsay and Rachel and others are investigating, is to take samples from patients at different stages of T1D to look at their B cell receptor sequences and how these B cell receptor sequences change in association with likelihood of development.
Oh, that's great answer. And then here's another one from Nita Mjal. Did you ever try isolating islets from these mice for GSIS?
We have not. That's something that we would like to do. We actually have paraffin embedded blocks, and I plan to do OTC media blocks because I would like to then do I plan to do immunofluorescence because we did look at insulitis, and despite the BCL6 deficiency, there is still insulitis in these mice, although it is lower, but it's not significant. I want to see what's different in terms of the immune cell composition in these mice and where they're localizing to see if that might give us a clue of why they're not developing type 1. It's a good question. Good suggestion.
Here's one more question. I'm not sure, but could you confirm that the anti-CD20 was not very successful in T1D, even though it shows protected in non-mice? Do you expect that selective treatment will have any effect?
Could you repeat the question again? I'm so sorry.
Kiara, can you
Yeah, there might be noise in the background. The question is regarding, like, I think the anti-CD20 treatment in humans was not very effective, even though the study showed that if you just knock out the all B cells from NOD mice, it show a very good protection. So my question was, when you're studying specific B cells in the mice, do you expect it would have better effect than targeting anti-CD20 itself?
I don't want to say better because I will say that what Rachel has done in the past is she's used a monoclonal antibody that will actually specifically delete the anti-insulin B cells in her mice, and we found that that was also protective. I don't know the rituximab literature as well, the anti-CD20 therapy, but I do know that I think there are many in the B cell field that wish they had tried it and say in a few other iterations of that trial, or maybe as a combinatorial therapy. I would yield to Dan on that, as I think he's more of an expert than I am, but I think there's more to be learned and done in the rituximab field to really get at why, what's going on and when's the best time to use it.
I mean, detailed sub-analysis of the rituximab data did show it's basically as effective as teplizumab, which is now clinically approved for stage 2, type 1. The stage 3 area is still difficult, and you have to normalize for a lot of features. But there's now a rituximab abatacept combined trial that I'm the national PI with Steve Gittleman on. So I don't think there's all hope lost for that. It's just using the right. I don't think we've had any singular therapy that's super successful, but finding the right combinations and tolerable combinations of treatments very important.
Yeah,
Thanks, Dan.
that's a good add value there. And the idea of the dual treatment, it looks like where everything is headed. One more question here. Any insights on the residual memory that B cells post-diabetic state could be reactive to exogenous insulin epitopes?
I would yield that I can happily give contact information to anyone to reach out to me. This is not my area of study, but I do know that in patients that receive exogenous insulin, those anti-insulin B cells do tend to persist. So I would expect that there probably is a greater skewing as you get more exogenous insulin, hence why we try to look at this pre-symptomatic stage before they receive exogenous insulin.
Okay, great. Thank you for fielding all those questions. There were, so many different types of questions. Very great talk and a great fielding of the questions. So I want to just ask Christopher Wilson to add slides, and we'll jump into this final presentation.
All right, great. So I'm glad that I got to go after Dudley because he gave a really nice synopsis of how we think B cells interact in type 1 diabetes. I'm going to talk a little bit about something that we're interested in our lab. And so Dudley focused very heavily on the idea that B cells activate T cells. And that, of course, is a very important aspect of type 1 diabetes. But what I'm going to talk about today is how B lymphocytes may actually erode the regulation that our body is so good at doing whenever we're healthy individuals. So most of us that don't have autoimmune disease, our body is constantly protecting us from developing autoreactive cells on a daily basis, because we pump out a lot of cells that could potentially harm us. Some of those are eroded and taken out of the regulation through central tolerance mechanisms, but our body is really good at peripherally controlling autoimmunity. And we think that autoimmunity breaks down, or protection breaks down in type 1 diabetes. So today I'm going to focus a little bit on the role of how B lymphocytes may disable protection in type 1 diabetes. So there was a question earlier about rituximab, and I very much agree with Dan. I don't think we know exactly how good rituximab could be clinically, because I don't think it's been applied correctly clinically. But rituximab did show efficacy when it was given. And so depletion of B cells, both in humans and mice, has shown some efficacy in beta cell protection. So we know that for certain. The other aspect that both Dan and Dudley discussed was that autoantibodies predict diabetes and your progression to diabetes. So the more diverse your autoantibody, auto-islet response, the more likely you are to develop diabetes. And we've actually staged diabetes by the number of autoantibodies we have, as well as your dysglycemia. So as Dudley illustrated, our current paradigm or thinking of how B lymphocytes work in type 1 diabetes is they're required to directly activate autoreactive T cells through antigen presentation. So just as Dudley showed you, those germinal center responses occur, and they seem to be very important for driving the destruction of beta cells. There are some challenges to that current paradigm that a lot of mainstream type 1 diabetes people have, both pointing to the rituximab trials and pointing to basic science in the NOD mouse, which is our best preclinical model for type 1 diabetes. So we know that if we genetically deplete or therapeutically deplete B cells, we can protect the NOD mouse from type 1 diabetes. But if we deplete the Tregs that sort of hold autoreactivity at bay, B cells are not required for type 1 diabetes to occur, for islet cell destruction to resume. So why is that? So if we think about the putative role of B lymphocytes in the anti-islet response in T1D, they could activate islet reactive effectors. And if we could knock those down, we could prevent or cripple islet rejection. If they inhibit protective Tregs, we can improve islet protection via Treg dependent mechanisms. So our main question is, do B lymphocytes impede Treg dependent protection of islets? And the secondary question, are B lymphocytes essential for islet rejection? So when I look at this, it's really hard to time and get enough NOD mice to look at where we all look at the NOD mice at the same time and see when they're going to get diabetes. It's all sort of stochastic a lot of times. So we take a different model. We take the NOD mice and we do islet transplantation. And what we can demonstrate is the anti-islet response in mice as we transplant them, and then we can follow them out until they reject those islets. So using that model, we took wild type NOD, we took B cell deficient NOD mice, and we transplanted them with allogeneic islets. So they will reject these islets regardless of whether they're treated with, well, they'll reject those islets regardless of whether they have B cells or not. So the NOD wild type reject the islets. The MUMTs will also reject the islets, indicating that B cells aren't necessarily necessary for rejection of islets. But when we treat with a tolerizing therapy called anti-CD45RB, this therapy seems to boost the ability of the body to do T regulation. We see that we can prevent islets from being rejected only in the case of the NOD MUMT. In the NOD wild type, if you look at this line, with the clear circle, you see that even with the treatment of this tolerizing therapy, we see that the NOD mice still reject these islets. When we look at the islet transplant itself, we can see in the NOD MUMT mice that accept the graft, they're surrounded by T cells, but those T cells seem to be of a regulatory phenotype, as indicated by CD25 and FOXP3 expression, which is a marker of Treg cells. So to sum up in that slide, what we're seeing is that when Tregs need to regulate the islet, B cells are impeding that process. They're impeding normal protective immunity from occurring, and islets are rejected, even when they're treated with a tolerizing therapy. So the next question we want to ask, if we take the NOD MUMT mice and we compare them to their wild type counterparts, and the only difference is one has B cells and one doesn't, are islet protective Tregs more abundant in those B cell deficient mice? So what we did in this case is we took Tregs sorted from NOD wild type mice and NOD MUMT mice, and we did TCR sequencing, and we used the MACPAS T1D related clones base to look and see the frequency of Tregs versus effectors in a MUMT mouse versus a wild type mouse. And the takeaway here is that for every effector that's targeted against your beta cell, there's more Tregs when B cells are not present, meaning you could have theoretically three effectors that are going to destroy the islet. And if they're in the MUMT environment, you're going to have more regulatory cells that can shut that response down. So B cells are impeding the frequency of Tregs in the type 1 diabetes model. So the next question we wanted to ask, are these Tregs functionally better? When we take them out of the B cell deficient mice, are they better at restraining type 1 diabetes? So we set up this experiment where we took Tregs from both our wild type B cell sufficient mice and B cell deficient mice, and we transferred them into immunodeficient NOD mice with diabetic donor splenocytes. And we monitored for hyperglycemia, just like Dudley described, checking blood sugars every week. And what we were able to see out of the 18 mice that received MUMT Tregs or B cell deficient Tregs, we had some that maintained diabetes, no diabetes at 100 days after the initial cell transfer, indicating to us that the Tregs that come from a B cell deficient environment are better at regulating diabetes than those that come from B cell sufficient environment. So to sum this all up, what we were able to demonstrate was the absence of B cells allows a Treg dependent tolerance to grafted islets to occur. So when there are no B cells present, you're better at tolerizing islets. It seems from our sequencing data that B cells limit the islet protective repertoire in NOD mice, and we're very interested to see if this occurs in humans as well. And if we increase the abundance of autoreactive B cells, this leads to a decrease in Treg functionality. So the more autoreactive B cells we have, in data I'm not showing, we demonstrated that if you have more insulin reactive B cells, you have less insulin reactive Tregs. And in terms of basic science, we want to look at when this occurs in the mice, where it occurs in the mice, and are there interactions that are occurring that we can target therapeutically to sort of boost the number of Tregs when B cells are present. So how are B cells and Tregs talking to each other, and can we change that so the Tregs are better able to do their job without B cell interference? So I did this work with Dan Moore. Dan graciously let me present it today, which I'm thankful for that. Dan, I did my PhD in Dan's lab, and now I'm a research instructor trying to spread my wings on my own here at Vanderbilt. And so I'd like to thank Dan and all the collaborators within our lab. We had some help from Immunomind, who helped us with the sequencing data and our funding sources.
Thanks very much, Chris. That was very interesting. And I'd be very curious to see what you're going to do next steps. How do you envision getting this towards a human trial? I mean, it's going to be tough sledding, right?
Sure, it definitely will be. I think one of the things that will be interesting to see is the abatacept rituximab combination trial that Dan's heading up. One of the consequences of this could be better Treg function. So hopefully at some point down the line, we can get our hands on clinical samples from that trial and see if Treg frequency or function is increased after a combinatorial therapy.
Yeah, that sounds great. If you can do that, that would be wonderful. I wanted to ask Al Powers, who's in the room, if he'd like to ask a few questions to the presenters or just give a bit of a wrap up.
Yeah, I wanted to thank Chris, Dan, and Dudley. Those were great. And the thing I liked about this session is it went from different immune cells to people with that. And so maybe I'll ask Dan the first question. Do you think you can connect any of your work related to the size of the pancreas to the immune abnormalities that Dudley and Chris spoke about?
I think that's a really good question, and I think I've been thinking about a lot. So, there's a pretty big distribution of the size of the pancreas, both in people with established type 1 and in our patients we've been following that are progressing to stage 3. We haven't looked at anybody that's gone from, no autoantibodies to stage 1. That'd be very hard to find and require imaging a lot of people. So I have a couple of models in my mind now. Our insulin gene mutation family suggests that the pancreas is shrunken a lot when beta cells are dysfunctional. If you think about type 1 diabetes and like the concept of endotypes, there's some concepts in literature that maybe some type 1 is a little more beta cell driven and beta cell dysfunctional. Some type 1 is a little more immunologically driven. I didn't get to show this, but we've also had the chance to image an individual with APS1, the AIR gene mutations. That's clearly very immunologically driven. His pancreas, he has type 1, or he has stage 2 type 1. He's not fulminant yet, but it's pretty normal size. So I'm starting to wonder if pancreas size maybe suggests whether you're more immunologic or more beta cell driven in your progression to diabetes. That'll take a lot of untangling to look at, but I think, between genetics and responses to various kinds of therapy and just following the pancreas as people move through progression may give us a lot of really interesting insights into what's the right treatment for people, when is the right treatment, and how they might be responding to treatment.
Dan, I had a quick question, just a sidebar here. Have you guys had a chance to look at any samples of pancreatic size in those who have ciliopathies?
So we haven't targeted individuals with ciliopathies, either that already have type 1 or that have it without having type 1. So we probably have 1 or 2 people in our big cohort who have celiac disease and type 1, but not enough to really make any comment.
Mm-hmm. Just curious. Thank you.
Could I ask another question? So Dudley and Chris, people hypothesized that in humans, type 1 diabetes is a relapsing, remitting, not a smooth process, right? Do any of your data with the mouse suggest that that might be occurring in your mouse model?
Chris, I don't know if you want to go first or.
Go ahead, Dudley. I'll let you.
I will at least comment, Al, that it's a good question. And in terms of the immune process, we do find that the germinal centers that we can detect in these mice that are at least insulin reactive do tend to be very transient. They tend to be active. You can detect them in mice and then they're gone. Same with the autoantibodies. They tend to peak and then they wane. And that might be due I would suspect that's due to the epitope spreading, that you're getting these other antibody or antigens are becoming activated and the target of the attack during the destruction. But I believe there is some evidence to suggest that the immune process does not exactly follow just a linear trajectory, but more of a roller coaster.
Yeah, I'm not sure. When I think of relapsing remitting diseases, I think of something like lupus. And I'm not sure whether it's relapsing or remitting, or if there's just a loss of the antigen it targets as the disease progresses. The beta cells are gone, insulin production is gone. Of course, there is exogenous insulin delivery. I don't know if it's relapsing or remitting, is more, it may be smoldering and just kind of slow progressing. I don't personally know of any evidence in mouse models that indicates a relapse and a remit session. But I think that it's probably a smoldering disease. Of course, you could think that Treg depletion is transient and the Tregs recover and then you deplete them again. So there's a lot of speculation here. But I'm not sure whether relapsing or remitting or sort of like a smoldering fire that takes a while to become a raging inferno.
Great. And then maybe I'll ask if I can ask one more question, Monica. I don't want to dominate.
Yes, of course.
So one of the things I liked about this session is that we had people using TrialNet samples. And so I thought that Dan Moore might comment on whether this is a common aspect of TrialNet, that samples are used for mechanistic studies. And if so, is there a way that that could be enhanced?
I would say what we've used TrialNet for is probably the most common approach where TrialNet's identified individuals kind of in a living biobank kind of situation, right? So the people come in for the MRI and we know what their status is because they've been in TrialNet, they've been identified, they've been enrolled, they've been found to have multiple autoantibodies, and then we can assess that individual. And I collaborate with Dudley and Rachel, and that's how we get samples for their studies that did the human profiling. This is not the samples didn't go to TrialNet and come back to us. We enrolled them while they were here because we knew that they were positive for TrialNet. So all those samples came from TrialNet individuals at Vanderbilt, as opposed to samples that had been stored and then unbanked. I think TrialNet is maybe a little bit more careful with studies, samples that have been in studies because they're just limited. So getting sample that's from a study where there was an intervention and then analyzing it outside of the already pre-planned TrialNet mechanistic study is a little bit harder. Usually you're not going to get sampled till all the other pre-planned assessments have occurred, but that does happen. Sometimes if you have a really well-established assay that has positive indicators in human studies. But the living biobank approach, you probably could draw nationally. We certainly could collaborate with other centers like we do on the MRI. That's a national collaboration, and we could ship blood around from those individuals. And if we had the right reasonable looking assay, we probably could apply it to stored samples, but those are small, right? There's just small freezers with small trial samples.
So Dan, let me make sure I understood what you said, that so TrialNet identifies the individuals and then through a separate protocol, Vanderbilt investigators collect samples that are in TrialNet participants, but not officially part of TrialNet. Is that right?
Because they go through what's called an ancillary study process. They're appended generally to TN01, which is the parent TrialNet study that's enrolling and identifying individuals with multiple autoantibodies. And then like our MRI study or Rachel's autoantibody study sits as an ancillary study underneath that. So there will be some reports back to TrialNet and some sharing of data with them, but generally the samples all then being handled locally for that, not going through like a central processing hub.
So I know the answer to this question, but I'm going to ask it anyway. So TrialNet doesn't support those studies by Dudley or Chris financially at all. Their infrastructure supports the identification of those. Is that
right? The dollars go to the infrastructure that provides identification. So there's an investment there, but there's not additional dollars for the MRIs or the sequencing or any of those things, some of which go unsupported.
Great. Thank you. That's all the questions I had, Monica.
Yeah, those are great qualifying questions, so we can really kind of fully understand the mechanics of the TrialNet and how those of you out there who are interested in accessing samples may go about doing it. So they're very helpful. Here's a question to Dudley McNitt. Has there been any further importance found in ectopic germinal cells found in the thymus that might contribute to autoimmunity?
Let me think about this question for a second.
It's in the chat to everyone.
I can speak on the pancreas where we do look for ectopic germinal centers in the pancreas, and they are
Germinal cell. Sorry.
Yeah, the ectopic germinal centers do form in the pancreas of these mice, and that's still to be determined about their importance. You can block them to some degree with like anti-CXCL13 antibodies, for instance, of previous work in our lab, but it doesn't necessarily have a huge impact on disease. In terms of what they're doing, that's still something we're trying to investigate, but they're clearly forming there, and it may be a site for the autoantibody production, but again, there's a lot that needs to be done about how important are they, at least in mouse models in T1D, and how are they driving the pathogenesis?
I can speak to I've thought a little bit about the thymus in terms of what B cells and B cell T cell interactions might be occurring in the thymus. So there could be that some of this, and we've thought a little bit about this, that we've seen abnormal B cell T cell interactions in the NOD thymus, and that this could be contributing to changes in T cell selection. So you're affecting the T cells at the earliest stage of their developmental trajectory, which could definitely have some sort of, peripheral consequence later on down the line, that the cells are more apt to be activated or less able to regulate. So that the the ectopic germinal centers or autoimmune response that's happening within the thymus could have profound impact on central tolerance of T cells.
Thank you. I'm not sure what the last comment meant. Well, thank you all for this excellent presentation, starting off with insights into T1D pathogenesis from the eyelid perspective. We had a great series of scientists talking us through that entire session with Marcela Brisova, Angela Cruz, David Jacobson, and Jian Cha, and then moderated by Danielle Dean, and then followed right on the heels with this presentation, insights into T1D pathogenesis, the immune perspective, with Drs. Dan Moore, Dudley McNitt, Chris Wilson, and Al Powers as the moderator. Just a great session with a lot of really wonderful questions, and we'll be posting this shortly on our website. Also, tomorrow, tune in for T1D prediction and prevention, a live fireside chat with Drs. Bill Russell, Dan Moore, and Al Powers, and they're really going to get to the heart of this sort of prediction and prevention problem, and how to best approach it, coming from their very long and excellent careers and historical wisdom. So thank you. And we'll see you again hopefully tomorrow. Have a great rest of the day today.